Hello, this is Matt Dean with A Plus College Ready, and we're going to talk about statistics in AP Biology today. If you look just down below my header there, you see two formulas from the AP Biology math formula sheet. The bottom one for standard deviation and the top one for standard error. Both of those equations often look very intimidating for AP Biology students. It's going to be our job today to make them less intimidating and to make sure that the students can not only calculate the two values, but also interpret their meaning. It's very likely that students will not actually be asked to calculate standard deviation and probably not even standard error on the exam, but they definitely need to know how to interpret the meaning of the values. One important point that we need to make is we need to make sure that students know the difference between two important terms, and those terms are population and sample. In statistics, when we're talking about a population, we're talking about an entire group of organisms. So maybe we're looking at the effect of fertilizer on the growth of apple trees. The population would be all the apple trees. Whereas the sample would be the apple trees that we've randomly selected to do our experimentation and measurement on. We're hoping that since we randomly selected them, that the sample is representative of the population. So next, let's talk a little bit about standard deviation. Standard deviation can be represented or abbreviated as ST, STD. It can be abbreviated as the lowercase Greek letter sigma, and that's if we're talking about a population standard deviation. Or it can be abbreviated as a lowercase s if we're talking about the sample standard deviation. That's what we will typically be dealing with in AP Biology. Standard deviation is essentially a measurement of variation, or sometimes dispersion, of a set of data values. It essentially compares how close each data value is to the mean of the sample. So it's, again, small standard deviations indicate that your data points are very close to the mean. You might say they're consistent. Whereas large standard deviations tell you that your data points are spread out widely on either side of the mean, more than likely. The standard error of the mean, on the other hand, can be abbreviated as SEM. You also might see it abbreviated as capital SE with a subscript X bar. So standard error, X bar, remember, is the mean, standard error of the mean. The standard error is an estimate of how far the sample mean is from the population mean. So we're comparing our small group of randomly selected data points to our entire population. Whereas the standard deviation is looking at how the individuals in the sample differ from the sample mean. So again, standard error is comparing the mean of the sample to the mean of the population. Here's what a sample problem might look like on an AP FR2. So you collected the following data in an experiment. During the experiment, you measured the height of plants grown at different fertilizer concentrations, 20 grams per liter of soil and 40 grams per liter of soil. The heights of 4 plants grown at each concentration were, and you can see the data. So we have 2 treatment groups. We've selected a sample size of 4 for each of those 2 treatment groups. The first thing we're asked to calculate is the mean for each group, then their standard deviation, then the standard error, and then we're asked to do some graphing and essentially then to interpret their results. So first of all, let's calculate the mean. As we said earlier, the mean is pretty much always in statistics symbolized as X bar. X with a line on top, that's X bar. So let's calculate the mean for the 20 gram per liter sample. We'll just put a little subscript here of 20. To calculate the mean, you're going to calculate it just like you normally would. You're going to add up all your data points. So in our 20 gram per liter sample, we're going to add together 18, 22, 20, and 26. And then to get a mean or an average, we're going to divide that by the total sample size, which is 4. So when we do that, 18 plus 22 plus 20 plus 26 divided by 4, for the 20 gram per liter sample, we get a mean of 22. And that's 22 centimeters. For the 40 gram per liter sample, and we'll do that down here, we're going to add together all our sample points, 32, 24, 29, and 23, and then we're going to divide that by the sample size of 4. So again, when we add 32, 24, 29, and 23, and divide that by the sample size of 4, we get a mean of 27 for the 40 gram per liter sample. The next thing we were asked to calculate is the standard deviation. Now here's our formula for standard deviation. So notice it says to take the square root of sigma, which means summation, of the quantity xi minus x bar squared over n minus 1. First of all, it's important to realize that xi is each of your individual samples, each of your individual sample data points. So for the 20 gram per liter sample, which we're looking at here, the xi's are 18, 22, 20, 26. You can see that notated here. So we're subtracting from xi the mean of the group. So for the 20 gram per liter sample, that mean is 22. The easiest way to do this, since you can't use a fancy calculator on the AP exam, is to build a table. So we're going to make our first column be xi. We're simply going to write in, which I've already done for you, all our individual data points, 18, 22, 20. Our next column is going to be xi minus x bar. So in this case, it's going to be 18, our first xi, minus our mean, 22. So that's going to give us minus 4. For the next one, it's going to be xi, 22, minus the mean for the group, 22. That's going to give us 0. For the next one, it's going to be xi of 20, minus the mean, 22. And that's going to give us negative 2. And for the last one, xi is 26, minus the mean, 22. And that's going to give us 4. Our last column in our table is going to be the quantity xi minus x bar squared. So that simply means to take this column and square it. So we're going to go minus 4 squared. We're going to get 16. Remember, when you square a negative, it becomes positive. 0 squared, just 0. Negative 2 squared is 4. And 4 squared is going to be 16. Now if we look a little closer at our formula, it says to take the summation of xi minus x bar squared. So that essentially means take this last column and sum it up, add it up. So 16 plus 0 plus 4 plus 16. If you add those up, 16, 4, 0, 16, you get 36. So our summation of xi minus x bar squared is 36. We want to divide that by n minus 1. N is our sample size, our number of data points. So we had 4 data points, 18, 22, 20, and 26. So n minus 1, 4 minus 1 is going to be 3. And then we need to take the square root of that whole quantity. So our standard deviation is the square root of the quantity 36 over 3, which ends up giving us a standard deviation, which we're going to notate as S, and we'll just write that here. S equals 3.46. All right. All right, so we just calculated the standard deviation of this sample to be 3.46. Now we want to calculate the standard error of the mean. Remember, standard error of the mean might be abbreviated as SEM. On your AP formula sheet, though, it's abbreviated as SE subscript x bar. The formula for it is the standard deviation divided by the square root of n, where again, n is the size of your sample. So in this case, we're going to take our standard deviation, 3.46. We're going to divide that by the square root of n. Our sample size is 4, so the square root of 4. So we end up with 3.46 divided by 2, which is what the square root of 4 is. So we end up with an SEX here of 1.73. Now these values are for our 20 gram per liter sample. So we need to do the same calculations for the 40 gram per liter sample. Alright, so now let's repeat those calculations for the 40 gram per liter sample. Our sample size again was 4, and for the 40 gram per liter sample, you can see our individual data points on this slide. They were 32, 24, 29, and 23. So the first thing we need to do is calculate the mean, which we already did on the previous slide, if you remember. When we calculated the mean on that previous slide, we got a mean of 27. So let's just write that up here. So the mean for this 40 gram per liter sample was 27 centimeters. So now let's work on calculating the standard deviation. So to do that, we're going to just follow our chart. Our individual data points, 32, 24, 29, 23. In the next column, we need to take those individual points, do them separately, and subtract the mean from each one. So here we're going to do 32 minus our mean, 27, get 5. Next we're going to go 24 minus the mean of 27 to get minus 3. Next we're going to go 29 minus the mean of 27 to get 2. And then finally we're going to go 23 minus the mean of 27 to get negative 4. Now we need to go through and square those values. So we're going to take 5 squared, get 25. We're going to take negative 3 squared and get 9, positive 9. 2 squared, give 4. And negative 4 squared. And we're going to end up with positive 16. Alright, so now that we have our chart filled in, now we need to take a look at our formula and calculate the actual standard deviation. So we want to find the summation of quantity xi minus x bar squared. So again, that means take this final column here, xi minus x bar squared, and add up all those values. So that's 25 plus 9 plus 4 plus 16. Again, when you add those up, 16, 4, 9, and 25, that gives you 54. So that is the summation of xi minus x bar squared. We want to divide that by n minus 1. Don't forget, n is the sample size. We had 4 samples, 32, 24, 29, 23. So n minus 1, 4 minus 1 is going to be 3. So we end up with 54 over 3. We can't forget to take the square root of that whole quantity. When you take the square root of 54 over 3, you end up with a standard deviation of 4.24. And that is our S value. Now we also need to calculate the standard error of the mean. Remember that standard error of the mean is equal to the standard deviation divided by the square root of the sample size. So for this calculation, our standard deviation was 4.24. We're going to divide that by the square root of n. Again, our sample size is 4. So 4.24 divided by 4, that's going to end up giving us a standard error of the mean of 2.12. So far we've completed part A. We calculated the mean for each group. We've also created or completed calculating the standard deviation for each group. We've also calculated the standard error for each group. The next thing we're asked to do is create a bar graph that illustrates the mean of the two groups to within a 95% confidence interval. And that means plus or minus 2 standard errors of the mean. So let's go to a graph. And on this graph, we want to go ahead and graph our data points. So the first thing that we want to do is label our axis. So we're going to graph, in this case, we're going to graph our fertilizer concentration. So we're going to go down here and we're going to label this as fertilizer concentration. Let's try that again. I'm going to do it over here on the side. I realize that should be my X label though. So that's fertilizer concentration. And you always want to include your units. So that was grams per liter. All right, now we only have two groups here we want to do. We want to do the 20 gram sample and we want to do the 40 gram per liter sample. So what I'm going to do is I'm going to count each one of these grid lines as 5. So this is going to be 5, 10, 5, 10, 15, 20. 20 right there. 25, 30, 35, and 40. So I'm going to graph my 20 gram per liter bar at 20 and my 40 at 40. I'm going to label those. So this one is 20. This one here is 40. You also want to go ahead and put in numbers for each one of those. So you wouldn't want to label 5, 10, 15, 20. I just don't have enough room for right now. And then over here on the y-axis, we're going to graph our mean height. Alright, I tried to clean up my graph just a little bit. Notice that I've clearly labeled my x-axis as fertilizer concentration in grams per liter. I've clearly labeled my y-axis as mean height in centimeters. Now something that I want to do that I'm not really able to do on this technology is to clearly label each of my tick marks, each of my grid lines. On the bottom, on the y, I'm going to make each one of these lines be equal to 5 grams per liter. And up on the y-axis, I'm going to make each one be 2. So what I want to do now is to go over to 20. So on my y-axis, 5, 10, 15, and 20. And I want to draw a graph, a bar graph, of the mean of my 20 gram per liter group. Now remember, that was 22, was the mean of that group. So I'm going to go here, and I'm going to draw up a bar graph up to 22. So let's go 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, and 22. So I'm going to come over here to 20, and I'm going to draw up a bar graph up to 22, like so. Alright, then I'm going to do the same for the 40 gram per liter sample. So that was 20, so this is 25, 30, 35, and 40. And I'm going to go up this time to 27, so this would be 24, 26, 28. So from 40, I'm going to go up to 27. Put me about right here. And then come down like so. Let's try to straighten that up just a bit. Maybe still a little off there. Alright, I'm going to go ahead and label that. Alright, so again, this is not pretty or not purely accurate, but for this one, X bar was equal to 27, and for this one, X bar was equal to 22. Now, the next thing we were asked to do was to calculate what's called a 95% confidence interval. And to do that, we're going to draw these things called error bars. We're going to have an error bar above the mean, top part of it, and the top part of that error bar below the mean. And let's go ahead and draw them first. To do that, we need to remember what our standard errors of the means were for both of those groups. So for the 20 gram per liter sample, our standard error of the mean was 1.73. So that was for the 20 gram sample. For the 40 gram per liter sample, the standard error of the mean was equal to 2.12. So to calculate or to create error bars, what we need to do is to go up 2 standard error of the means from the mean, and also go down 2 standard errors of the means. So for the 20 group, standard error was 1.73. Twice that ends up giving us 3.46. So we need to go to the mean, we need to go here to 22, and we need to go up 3.46. So that would put us at 25.46. So we're going to start right here at the mean, and we're going to go up to 25.46. That'll put us somewhere right in here. We're going to draw this little crossbar. We're also going to go down 3.46. So if we take 22 minus 3.46, that gives us 18.54. So we need to go down to 18.54. So we're at 22 at the mean. So if we go down 3.46, somewhere right in here. So those are our error bars. What those error bars tell us is that between the top part of the error bar and the bottom part of the error bar, there is a 95% chance that the population mean lies within that range. So again, we created a 95% confidence interval. There's a 95% chance that the population mean for this particular population lies between this point and this point. Let's do the same thing for our 40 gram per liter sample. So our standard error of the mean for that one was 2.12. We want to multiply that by 2, so that puts us at 4.24. We need to go up from the mean 4.24. So if we take 27 and we add to 27 4.24, that gives us 31.24. So we need to go up to 31.24. So let's see, that's 28, 30, so that's going to put us somewhere in there. All right, we also need to go down 4.24. So let's take 27 and subtract from 27 4.24. And when we do that, we end up with 22.76. So now we need to drop down to 22.76. So that's going to drop us down to somewhere in here. So now we're saying there's a 95% chance that the population mean for the 40 gram per liter sample is somewhere between this point and this point. So if we back up a bit and we look at the final part of the question, the final part of the question said, based on the sample means and the SEMs, are these two populations or groups significantly different in terms of height? Did the fertilizer affect plant height, and justify your answer? when you get this type of question, which seems to show up on every AP exam lately at least, you want to look and determine if the two error bars overlap for your two groups. Notice that right in this area, the error bars overlap. What that tells you is that the population mean for this group and the population mean for this group could lie somewhere in this area. They could be the same. So what that tells you is that they're not significantly different. If the error bars overlap, the two samples are not significantly different. And in this case, that tells you that the fertilizer didn't really make much difference. So here I went ahead and graphed the two sets of data with Excel. And again, I just wanted to show you that the way you determine if these two groups are statistically different or not is you look at their error bars. And just like we saw in my rough hand-drawn sketch, right in this area, the error bars overlap. And that tells you that these two samples are not statistically different. It tells you that the population mean for this group, the 20 gram per sample group, could be exactly the same as that for the 40 gram per liter sample. Again, if the error bars overlap, the two groups, the two samples, are not statistically different. And in this case, that means the fertilizer had little, if any, effect. Here's an FRQ that deals with error bars and standard errors of the mean. This came off the AP exam just a few years ago. It says, trichomes are hair-like outgrowths of the epidermis of plants that are thought to provide protection against being eaten by herbivores. In a certain plant species, stem trichome density is genetically determined. To investigate variation in stem trichome density within the plant species, a student counted the number of trichomes on the stems of 6 plants in each of 3 different populations. So in each case, we had a sample size of 6. The student used the data to calculate the mean trichome density for each population. The results are provided in the table below. So again, notice we have 3 populations. We have a sample size of 6 for each population. Here, they calculated the mean for us, and they did that work for us. And here they calculated the standard error of the mean for each one. Now, the standard error of the mean for all 3 of these was 1. In reality, that's probably never going to happen. It did in this case. So it asks us, on the axes provided, create an appropriately labeled graph to illustrate the sample means of the 3 populations to within a 95% confidence. So we want to graph it, just like we did a second ago. So on our graph, we want to put the population number on our x-axis, and we want to put the mean trichome density on the y-axis. And I want to abbreviate a little bit. And we also always want to put the units. So in this case, it's trichomes per, trichomes we're looking at trichomes per centimeter squared. Alright, so I want to make each one of my x-axis units be equal to a half. So that means this would be 1, 2, 3, 4, 5, and so on. And the mean trichome densities, I'm just going to make each one of these lines be equal to 1. So we'll be 1, 2, and I'm not going to label all these. You would want to make sure you label all these. I'm not going to label each one right now. So we want to graph the means with a bar graph. Population 1, the mean was 9. So we'll go over to 1 and we'll count up to 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Make a little mark there. And then we want to draw our bar down. Again, that's very messy. I'll show you a more professionally done one of these here in a second. But that's X bar is 9. For population 2, our X bar was 11. So we're going to go up to 11 at 2. Put us here. Draw down our bar like so. And then for population 3, X bar was 14. So let's see, that puts us at 12, 13, 14 right here. Boy, that's terrible. Let's try to clean that up a bit. All right, now we're also asked to create the 95% confidence interval. And remember, that means take our standard errors of the mean, go up 2 standard errors of the mean, and down 2 standard errors of the mean on each side. So for population number 1, our mean was 9. We need to go up 2 standard errors, which would put us at 11. And we need to go down 2 standard errors, which would put us at 7. So there's our error bars for population 1. And again, that means there is a 95% chance that the actual mean of the entire population lies between 11 and 7. For population 2, our mean was 11. Our standard error of the mean was 1. So we need to go up 2 standard errors of the mean. So that means go up to 13. Draw down an error bar. And we also need to go down 2 standard errors. So that means go down to 9. Again, put an error bar there. So that means there's a 95% chance that for population 2, the population mean is somewhere between 13 and 9. For population 3, our mean was 14. Our standard error was 1. That means we need to go up 2 standard errors. So we need to go up to 16. We also need to go down 2 standard errors. So that means we need to go down to 12. So that tells us for population 3, there's a 95% chance that the population mean is between 16 here and 12 here. Now if we look at the last part of this question, the second part of this question, says, based on the sample means and the standard errors, identify the 2 populations that are the most likely to have statistically significant differences in the mean stem trichome densities, and justify your response. Remember, we tell how different they are by looking at the overlap between their error bars. Notice that group population 2 and population 3, they overlap in here. That means that those 2 groups are not statistically very different from each other. Also notice that group 1 and group 2, they overlap in that area, so they're not statistically very different from each other. But notice there is no overlap between 1 and 3. So our answer to part B, populations 1 and 3 are the most likely to have statistically significant differences. And we're going to justify that by saying that their error bars do not overlap. That's all we need to say on part B. Now the last part of the question we'll come to here in just a second. Here's a version of the same graph I just drew by hand. This one was done with Excel. Again, you can notice that groups 2 and 3, their error bars overlapped in this particular area. Group 1 and 2, their error bars overlapped in here. But again, there was no overlap between 1 and 3. Therefore, groups 1 and 3 are more likely to be statistically different than any of the other 2 groups. Now, part C wanted us to describe the independent and dependent variables and a controlled treatment for an experiment to test the hypothesis that higher trichome density in plants is selected for in the presence of herbivores. The question wanted us to identify an appropriate duration of the experiment, and it also wanted us to predict the experimental results that would support the hypothesis. What I want you guys to get used to looking at are the rubrics for each of these FRQs. You can find those online. this is the actual rubric for this particular question. Let's go ahead and look at parts A and B first. Notice that on part A, you could earn a maximum of 3 points. You could earn 1 point for correctly labeling, scaling your graph with proper units. You could earn another point for drawing the bar graph and plotting the means correctly. And you could earn a third point by correctly drawing in the error bars. For part B, you could get 2 points maximum. 1 point for identifying that populations 1 and 3 are the most likely to be statistically different. And then another point for essentially saying that that is true because their error bars do not overlap. Now for part C, again, we're describing an experiment to test the hypothesis that higher trichome density in plants is selected for in the presence of herbivores. So this particular part of the question was worth 5 points. You could get 1 point for identifying your independent variable. Now there were a couple of ways to approach this part of the question. The way that I would have approached it, I would have said that my independent variable is the presence of herbivores. Remember that your independent variable is the thing that you are intentionally manipulating and changing, the thing you're trying to test. So we want to know the effect of the presence of herbivores. The dependent variable is the thing that changes because of a change in the independent variable. So our dependent variable would be the amount of trichomes, the trichome density. A good control would be to use the same kinds of plants, but no herbivore, no presence of a predator. Appropriate duration. An appropriate duration would be several generations. So as long as you said more than 1 generation, you got credit on this. And a good prediction would be that due to the presence of herbivores, the trichome density would increase. So again, there are other ways you could have earned these 5 points, but I think this is the most straightforward way to do so. So this particular question was worth 10 total points, 3 for part A, 2 for part B, and then 5 down here on part C. And you really only needed to know how to do the statistics really on just part B. Alright, here's an additional question that dealt with error bars, and this was an FRQ off of an AP exam from several years ago. It says adult male guppies exhibit genetically determined spots, while juvenile and adult female guppies lack spots. In a study of selection, male and female guppies from genetically diverse populations were collected from different mountain streams and placed together in an isolated environment containing no predators. The study population was maintained for several generations in the isolated area before being separated into 2 groups. One group was moved to an artificial pond containing a fish predator, while a second group was moved to an artificial pond containing no predators. The 2 groups went through several generations in their new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined as shown in the figure below. Vertical bars in the figure represent 2 standard errors of the mean. Alright, so what I want you to notice is that here at the beginning, the error bar is very, very big. So remember, if the mean is 10, right here, we went up to 12. So that should tell you that the standard error was 1, because you got 2 standard errors. Close to 1 at least. Notice that over time, those error bars tended to get smaller until here at the very end. So part A. Describe the change in genetic variation in the population between 0 and 6 months and provide reasoning for your description based on the means and the SEM. So once again, let's go to the rubric. The grading rubric for part A. Remember, in part A, we're trying to describe the change in genetic variation between 0 and 6 months and provide reasoning for this description based on the means and the SEM. So if we go back to our graph for a second, here we started off with error bars that were very, very far apart. And over time, those error bars got smaller, at least between 0 and 6 months. So because the error bars were smaller, that meant that the standard error of the mean was smaller, which meant that the standard deviation was smaller, which meant that the variation in those samples were smaller. So what we needed to say here, in terms of the rubric, was that genetic variation is decreasing between 0 and 6 months. And we're going to provide a reasoning essentially by saying that the standard error of the mean gets smaller between 0 and 6 months. Now part 2 of the question, or part B, propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 20 months. In the absence of the predator. So we're looking about in the absence of the predator. So in this case, we're looking, this is the group for absence of a predator. And we notice that between 6 and 20 months, so somewhere between here and here. So we're essentially looking at this part of the graph. And what I'm hoping that you notice is between here and here, the number of spots is increasing. So what we want to say for B, propose one type of mating behavior that could have resulted in the observed change in the number of spots. So let's go back to the rubric. All right, and the rubric for part B says, most importantly, the number of spots probably went up due to sexual selection. Females are attracted to males with more spots. So saying that sexual selection for increased spots is happening. You might have also explained this by saying that random mating, that the number of spots increased essentially due to genetic drift, due to chance. Either one of those would have been appropriate answers. Sexual selection is probably the best answer on part B. Also notice part C says propose an evolutionary mechanism that explains the change in average number of spots between 6 and 20 months in the presence of the predator. So let's go back to our graph. So again, 6 months to 20 months. This is the error we're looking at. This time we're looking at the predator present, which would be this part of the graph. And notice that essentially we have a line going down. That tells us that the number of spots is decreasing from 6 to 20 months when there's a predator present. So let's go back to the rubric. And what the rubric here is saying is that this is probably due to predation, that the predators see the spots. That allows them to identify the fish and prey on them, to eat them. So here we're saying that there's directional selection against individuals with a lot of spots. Those individuals are getting eaten. Or we could say that kind of in another way, that individuals with fewer spots are being selected for. We could describe that in terms of natural selection. We could say that individuals with fewer spots survive longer because they're not eaten as often. We could also maybe explain this in terms of genetic drift, that these changes happened by chance. But what they wanted you to go for mostly on part B was to talk about part B being about natural selection, part C being about natural selection, whereas part B was mostly about sexual selection. So once again, this FRQ was worth 4 points, 2 points for part A, 2 points maximum on part A, 1 point maximum on part B, and 1 point maximum on part C. And again, really the only time you had to understand the statistics, or at least in this case the error bars, were on parts A, really. That's when you needed to understand how error bars work. So I hope that helped.